Hello, my name is Caroline Jones, and I am a new assistant professor in the Department of Bioengineering here at UT Dallas. I want to thank you for giving me this opportunity today to talk to you about the research I do in my laboratory of immunoengineering and therapeutics. First, I would like to thank my amazing team. You can see by my current and past lab members that not only do I recruit successful graduate students, but also creative, hardworking, and innovative undergraduate researchers as well. I'm always looking to recruit more hardworking undergraduates to my lab, so if you're interested in some of the research topics that I talk about today, please email me at caroline.jones@utdallas.edu. Working in my lab as an undergraduate researcher takes about a 15-hour per week time commitment, but you'll learn a lot of very useful technical skills and engineering skills working directly with a graduate student on one of the engineering topics that I described today. This is the gorgeous view out of my second floor office in the Bioengineering and Sciences building. What drew me to UT Dallas was not only the incredibly strong engineering department that was originally developed by Texas Instruments and the clean room that enables me to fabricate my microfluidic chips, but also the close ties and collaboration with UT Southwestern. This is really the perfect environment for a bioengineer because we can develop engineering tools and then take them directly to the patient bedside at UT Southwestern with the goal of improving human health. So what do we do in my laboratory? We focus on technological innovation and creating and engineering novel microfluidic platforms to measure with single-cell precision immune cell function. We can use these chips to, one, investigate the biology of inflammation and very specifically define the microenvironment of immune cells to investigate how the microenvironment or epigenetic reprogramming of innate immune cells influences function. We can also take these tools directly to the hospital setting and do studies on septic patients and understand how immune signatures correlate to specific disease states. So all of you are healthy undergraduates, but I know that you're familiar with going to the physician and getting panels of biomarkers, for instance, looking at your cholesterol or even your blood cell counts. And currently, physicians will use these white blood cell counts that if they're too high, that can indicate infection, or too low, then the physician needs to investigate that further. And the idea from my lab is that not only will we get the counts of white blood cells, but actually how the cells are functioning. Because if you have cells, but they're not functioning properly, they really won't help in fighting off pathogens, and they can even cause damage. And really, in these unusual circumstances, I don't need to convince you that the immune system is important. Inflammation plays a role in every single disease. I look at sepsis, which is the overreaction of your immune system to infection, but inflammation also plays an important role in cancer, this current COVID-19 pandemic, cardiovascular disease, and it even plays a role in neurological disorders such as Alzheimer's disease or depression. In my lab, we make microfluidic chips where we can define precise gradients of chemicals and then look at how the cells migrate on these chips. And some of you may be familiar with transwell assays where you put a chemical signal on one side of a membrane and the cells on the other, and then you count the percentage of cells that migrate. But with microfluidic platforms, you get a lot more quantitative data. So you can look at how single cells migrate, which is important if there's heterogeneity in the cell populations, but you can also get more high resolution data and temporal data about how the cells are migrating, including their velocity, directionality, and whether they oscillate. So here on the left, you can see one of my microfluidic chips on a glass slide. And in the center, these are my neutrophils. That's your most abundant white blood cell migrating towards a linear gradient of chemoattractant. And on the right, you can see me in a suit. And you may think this is because I am handling pathogens, but actually in a clean room setting, we need to make sure not to get any dust particles on our microfluidic chips. And this is because these small channels are less than the size of a hair. So they're smaller than the cell. They're on the scale of microns. And so we have to be in this clean room with very few particles in the air to enable fabricating these devices. And how do we get this data? We take microscope images every minute or two, and then we can track how the cells move using image processing software or machine learning. I specifically look at sepsis. So that's when your immune cells overreact to an infection. And it's a life-threatening response of the body's immune system to an infection, which can potentially lead to tissue damage, organ failure, and death. It's the leading cause of death in hospitals and intensive care units, and it can cost hospitals around $20 billion per year. And so we ask a lot of questions about what the role of innate immune cells is in sepsis. How do immune cells migrate and prioritize which chemoattractant ingredients to follow in these complex tissue microenvironments? And does prior levels of inflammation or pre-existing conditions influence your immune cell function? And this is a simplistic view of some of the experiments that we run. We can collect the patient blood at UT Southwestern. We can run that on a microfluidic platform, and then we can look at some of these phenotypes that we see in septic patients and not in healthy donors. And then we can use this to predict whether a patient has sepsis. So from this data, you can see that there are some markers of neutrophil activation that are able to even diagnose sepsis before the physician, several days before that. And so that's critical in monitoring and using it for an early warning sign in sepsis. We can also ask basic biological questions and be able to target this with drug therapies to be able to prevent sepsis in the future. So one of the questions that we asked about a biological question was we made a second iteration of our microfluidic device where we put cells in between two different signals. So let's say the green signal is a good signal, that's where the cell should go, and the red signal is a bad signal or an inflammatory signal, and the cell should prioritize the green signal over the red signal. We showed for the first time that actually the epigenetic programming of cells or the microenvironment they were in before we put them in a microfluidic device can tell them which direction to go or can tune them to migrate one way versus the other. So with a high dose of LPS, cells migrate in a high abundance towards the FMLP or the high signal or the green signal. And if we prime them with a super low dose of LPS, they actually can migrate the other direction towards the inflammatory signal. And what's really cool about working in my lab is that not only we work with bioengineers, but we also collaborate with world-renowned immunologists and mathematicians. Here, Professor Lee and Professor Chupe are both collaborators that I have from Virginia Tech, and they can help us to answer this transdisciplinary big problem question of sepsis. So here are two movies that my graduate student Brittany took, and you can see here that these are healthy cells. You can see them migrating along the channel. These blue cells are migrating along the channel in both directions. But then when we prime them with LPS, actually more of these neutrophils or these blue cells migrate towards the inflammatory signal. And I can look at these gorgeous movies all day long, but we can quantify them using machine learning or image processing techniques, and we can get a lot of quantitative data about the percentage of cells that migrated, the rate of accumulation, and even the velocity of these cells. And then we can use that data to be able to create mathematical models about the receptors and the kinases that determine these phenotypes. And then we use these microfluidic data to feed this model. We can solve the model, and then we can make future predictions about how these cells will perform based off of priming or tolerance. And then we can reiterate this process over and over. So we use microfluidic platforms, these wet bench experiments to inform our model, and then we check our model, and then we can improve upon that until we get the answers to the basic biological questions that we're looking at. And then a lot of the experiments that I've shown you so far simply take chemicals on the chip and look at how the cells migrate. But not only can we put chemicals on the chip, but we can actually put the pathogen on the chip. So here, these red hyphae are fungal cells, and you can see that they're branching and growing in these microfluidic platforms. And Aspergillus is a fungi that's opportunistic, meaning it's all around us, and normally a healthy immune system can fight it. But here you can see on this chip that the neutrophils only started migrating to the microfluidic platform after the hyphae started growing. So here you see that these small dots of conidia, they start to hyphae, and then that's when you bring in the neutrophils. But if we prime this, let's say with an immunotherapy, and really drive the immune cells to migrate towards the chip, we can bring them in much quicker. So here, with this immunotherapy that I'm testing, or stimulating them with a certain signal, you can see that the cells are able to swarm and phagocytose the Aspergillus and kill it. What's great about microfluidic devices is that we only use a small droplet of blood, and that we can run combinatorial or many experiments at once to be able to test drugs. And so we can test hundreds of different immunotherapies and see which one produces the immune response that we want. Not only do we look at migration, but we can look at other killing mechanisms. Neutrophils release their DNA. This is called a neutrophil extracellular trap, and it actually will trap the pathogen. So my student here on the upper right, this is her at BMES. She designed a well device where we can co-culture neutrophils with Pseudomonas and look at the timing of the net release and do immunostaining to see specific markers of these nets to see if there's any heterogeneity in the nets. My other student, Nidhi Menon, she looks at tuning the microenvironment. So here, her work was recently featured on the cover of Biomaterials Science, and she used different hydrogels to make a three-dimensional microenvironment that presented different growth factors to spheroids of breast cancer, and then ran RNA-seq studies to see how this changed their expression levels of different genetic markers. And she showed that hundreds of markers were changed just dependent on the microenvironment. She also developed a biosensor to look at specific cell adhesions, to look at ICAM, which is a cell adhesion molecule. And what's cool about this engineering biosensor is that it's in real time. So instead of just doing an endpoint immunostaining, she can look at the real-time expression of ICAM. And finally, my graduate student, Dr. who just graduated and is now a postdoc at Virginia Tech, she developed a microfluidic device where she could apply a low intensity electric field to drive drug delivery towards cancer cells. And what's really cool is she took this project a step further, and this was featured on the back cover of Lab on a Chip. What's really cool about her study is not only did she use the electric fields to delivery, but she also showed that you can use the electric field to herd or to drive immune cell migration as well as drug delivery. So look for that. That's a paper that will be coming out very shortly from my lab. And then finally, I'm sure you can tell by this study that I really love immune cells. And so we've also done some outreach programs where we can use a virtual reality program to teach high school, middle school, and even college students about immunology. And what's cool about this, and most of you are now in a virtual learning platform, is that it can be very interactive. And so it's really like playing a game, and you can learn a lot more about immune cells playing this game. And so we've collaborated with some artists, and we hope to do some outreach to get this STEM material out to middle and high schools. So that's all I have for you today, and I really appreciate you watching this whole YouTube video. And please reach out to me if you have any questions about my research or you're interested in working in my lab as an undergraduate researcher. Again, my email is caroline.jones@utdallas.edu. Thank you, and I hope you have a wonderful day. And stay safe and healthy. Bye.